This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 511, recorded on September 14th, 2018. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me here today in New York City, Dickson Despommier.
Hello, Vincent.
Cloudy out.
It is.
Foggy, rainy.
It's 70 some odd degrees, and it's like 60% humidity.
Very humid. It's been raining like for 10 days now.
Yeah,
right?
off and on.
But there's this hurricane which just hit the Carolinas.
Yeah, they were lucky. That thing downgraded really fast.
It did?
It went from a 4 to a 1.
Nice.
In less than 24 hours.
Well, that's good.
Yeah, but it brings all of the rain and all the surge with it.
I understand. Well, my friend Michael Schmidt, who does TWiV, he said the university's been closed since Tuesday.
That's right.
It's terrible.
That's right.
Probably there's no reason to do that.
Well, there was when they told him to close it.
Also joining us from Madison, New Jersey, Brianne Barker.
Hi, it's great to be here. It is similarly 73 degrees here, or 23 Celsius, and gray.
Maybe it'll stop being gray next week. Who knows?
That'd be nice.
No, Saturday and Sunday is supposed to be something.
Nice.
Yep.
Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. How you doing?
Okay.
We have 81 degrees and cloudy with occasional rain showers. We've finally broken our dry spell. We've had rain for about the last week or so, and it's great. Everything's greening up. It's I think we're out of the 100-degree stuff, so we're back into something more reasonable weather-wise.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here. Glad I wasn't totally forgotten. Thank you, Dickson.
No worries.
It is equally gray here. A couple of degrees colder. It's 71 Fahrenheit, 22 C, and 78% humidity, and just gray.
78.
Oh, that's a lot.
Yeah.
Right.
All right. So our special guest in studio comes from New York University Langone Medical Center, Ned Landau. Welcome to TWiV.
Hi, thanks, Vince. Good to be here.
Now, is Langone the Home Depot guy?
That's the same guy. That's him.
Okay.
Oh.
Wow.
You get your name on a medical center. Dixon, you could get your name on a medical center.
Not really.
You might have to first earn a few $100 million and then give it away.
Even if I did it so long that we'd never put it up there.
So it's interesting that you're here today because last week, the episode I posted was the one recorded at Cold Spring Harbor.
Oh, yeah.
Way back in May.
I was there.
Where I saw you.
Yeah.
And I said, you should come on TWiV sometime. And now here you are.
Yeah.
It says that's how long it takes, right?
Oh, okay.
From May to now.
So Ned was in the audience when I was there.
He was at the meeting, and
I
saw him at the bar afterwards.
Cool.
I was just wiping up the top of the bar, and
There was a lot of people at that bar.
Well, and it takes a while to get somebody over from NYU Medical Center because across town and, back
Cross town
up.
is very hard.
Well, no, it's an A train. Come on, you hop on the A
Or
train.
you take the
It was
M4
okay.
bus.
Did you
That
take a
takes me.
taxi?
I took Uber.
Uber.
Oh, Uber.
Okay.
Oh, okay.
Yeah, that's fine.
I got here fast. There was not much traffic today.
Yeah. All right. I want to remind everyone before we start to give some money to the Viruses and Cells Gordon Research Conference, 2019 conference, which is being run by Julie Pfeiffer and Britt Glounsinger. They need money, $70,000 to support partial registration and travel for 32 speakers, 18 discussion leaders, and selected students and postdocs. We'll have a link to the donation site in the show notes, and you can go and give whatever you want. Help
And you
them
should
out.
give money to the conference because if you give it directly to the viruses and the cells, they'll just blow it.
It's true.
Parasitize it.
Yeah.
And one more thing. Martin writes, congratulations on the 10th anniversary this month, September 2018. Of course, that's the anniversary of TWiV. What precise date might that be? Martin, Patreon contributor.
Nice.
So TWiV number 1 was released on September 24th, 2008.
Outstanding.
Dickson and I recorded number 1, West Nile virus.
Yep.
Do you remember?
I do. Very much so. And not here either.
And this, of course, today is not the exact date, but the 24th is...
Close enough.
It's a Monday, I think.
Is that a Monday?
Yeah.
Yeah.
It is. It is. And I will be in Belgium.
Uh-huh.
And interestingly, the 24th of September is also the birth date of our first son.
Oh,
So he was already born at that time. Otherwise, I wouldn't have been allowed to podcast if that was his birth date, right?
Right.
But yes, so it's an interesting date. 10 years. And of course, on this 10th year, we did our 500th. So
yes.
we're kind of in sync. So thank you for reminding us, Martin. All right. So, Ned, we're going to talk about some of the work that you do. And of course, it's on a retrovirus, HIV-1. And, but before we do that, I wanted to hear a little bit about you. Where are you from originally?
I'm from the great state of New Jersey,
Wow,
Bergen County,
Bergen County?
Teaneck, New Jersey,
Oh my gosh.
just over the George Washington Bridge.
I grew up in
Very
Dumont.
good.
Steve lives
Oh, really?
in Tenafly.
Well, I live farther
So
away.
this
But I
is
was
a
Just
whole
still in
New
Jersey.
Jersey show here.
I was born in Patterson.
So was I.
No, what hospital?
I don't remember you.
Yeah.
You took different exits.
How could you not remember?
I don't know the name of the hospital, but yeah, my mother was from Patterson
Yeah.
and her family. Yeah.
It's interesting. I didn't know that. Brianne, are you from New Jersey?
I am not from New Jersey. I'm from the middle of nowhere, upstate New York, but I'm in New Jersey now for a while.
Where are you from? Wells, Oswego, or some of those?
Town is called Camden.
Camden. Okay.
Yes.
Yeah, that's the confusing one, which is supposed to be in New Jersey, but
There's one in Maine that's quite lovely.
Camden, Maine.
Camden,
Yeah,
Maine.
it's lovely.
And so you went to school in New Jersey? Is that right?
I went to high school in New Jersey,
In Tenafly.
in Teaneck High School.
Teaneck.
Great high school, by the way.
Yeah.
And well, I spent most of the time sitting outside. I think, from what I recall.
Really?
We had a lot of fun sitting around. outside.
By definition of you wanted to do that, or someone asked you to sit outside.
No, we just had spare time. We're not that interested in some of the classes, and
Oh.
had a lot of good friends.
Were you just in science at that time?
No, I was not. I was interested in English literature. I didn't get interested in science actually till I was in college, and I took a microbiology class.
Nice.
And I started to learn about immunology through that class. And it really fascinated me because I could not understand how you could be immune to any conceivable molecule, or your antibodies could be raised to any conceivable molecule, and how that would not take up all of your genes in the whole human genome. I thought immunology would be a good field to get into, and that it was amazing that with, just on that little bit of intuition, that turned out to be very good idea.
Now you know how that works, right?
Yeah, well, after college, I went out, I hung out, I took a year off.
Where'd you go to college, by the way?
I went to Grinnell College in Iowa.
Ohio.
Oh, that's a
Iowa.
Iowa, right.
I took a year off. I didn't know what to do. I went out to Berkeley. I sat in some of the classes at UC Berkeley.
Uh-huh.
And Susumu Tonegawa came and talked.
Ah.
And he said, we've been trying to figure this out, and we see that immunoglobulin genes can rearrange.
Mm.
And I thought, wow, that is just an amazing finding. And so that's how I got interested in immunology, and then decided to go to grad school, or went to MIT.
That's where I met you.
That's right.
You were a grad student with David Baltimore.
I was in the Baltimore lab,
Right.
and I remember you as one of the senior guys that we looked up to. I was as a young grad student.
So I remember that you cloned terminal deoxynucleotide transferase.
That was
correct?
That was my project, my
Yeah.
thesis project. Yeah.
Because I used TDT, which was not cloned. We had
Right.
to get it, to put tails on plasmids and inserts to clone polio DNA, basically. That's how we cloned back then. You would make DNA, cut your plasmid. We used PST, and then you could do GC tailing with TDT to get it in there.
That's right. Everybody did for making cDNA libraries. TDT was used, but when I was in grad school, David Baltimore was wondering about, what is the real function of this enzyme? Because it's not just there to make so that you could make cDNA.
right.
And he thought, well, maybe it's a way to get diversity in immunoglobulin genes by putting on a few additional bases.
Yeah.
The enzymatic activity was to add random nucleotides onto the end of
Right.
DNA. And so in order to do those studies, we had to clone the gene. And we didn't have any way to clone the gene for it at the time. We didn't know anything about it. The only thing we had was an anti-serum against the protein. And at that time, Irv Weissman out at Stanford was trying to come up with a way to clone genes with using antibodies. So make an expression library and then screen it with antibodies. And it hadn't really worked. But David sent me out there with the anti-serum and said, hey, go to Irv's lab and try this. I went there for a few weeks, and sure enough, I got the clone.
Nice.
Wow.
And
Nice.
so that was my thesis project. And then
Did it have to do with antibody diversity at all?
yes, and that turns out, so his idea turns out to be the correct idea. And so I published the first paper showing that did seem to be real, that you could get a few bases added to immunoglobulin genes from TDT. And then Fred Alt and others showed that it really does, it creates these so-called the D region of the immunoglobulin genes. The diversity region is made by terminal transferase.
Cool.
So Fred Alt was in the lab at the time. Steve Goff was in the lab at the time. Speaking of Fred Alt, I just listened to a podcast this week with his son,
Yeah,
Jamie Lopez Alt. He's a famous chef, apparently.
he is.
Yes, he is.
It's amazing. And so he opened a restaurant in San Mateo, and he talked about how it was a nightmare to do this, and I'm thinking, this is incredible. It's Fred Alt's son. I didn't know that until
And what's funny is that his thing is the science of cooking.
Yes, that's right.
And so obviously it's
Came
got from his dad.
Yeah, it's exactly right. It's exactly right. Wow. All right, so then you did a postdoc where?
I was a postdoc with Dan Lippman at
Right.
UCSF.
Oh, in UCSF.
Yeah, before he was at NYU.
Because he moved afterward?
Yes, he moved after I finished.
Okay.
And
What did you work on there?
that's when I first started working on HIV. So we started working on just interactions between the HIV glycoprotein GP120 and CD4. That was my main project, and we were understanding how HIV incorporated its envelope glycoprotein into the virus particle. So there's this phenomenon called pseudotyping, where viruses can take onto themselves the envelope glycoprotein from the wrong virus, from a different virus. So we were studying how that whole process worked.
So Dan was a postdoc here, right, with Richard Axel.
That's correct.
So all this stuff is all tied together, right?
They're all genomically linked.
I think he'd identified CD8, right?
And CD4.
He did 4.
In the Axel lab, yeah.
And but the other guy in the Axel lab figured it out as a HIV receptor, no? Who was the other fellow at the
Paul Madden.
Paul Madden, right?
Yeah.
He was a student. All right, and then you finished and you came to NYU initially, is that right?
No, so I when I finished up in Dan's lab, I became an assistant professor at a brand new AIDS research center in New York City called the Aaron
That's
Diamond
right.
AIDS Research Center. I was there for about 7 years, and then took a little detour to Southern California, went to the Salk Institute, and then came back again to New York City, to NYU.
So we just had a microbiology retreat on Friday, and David Ho gave a talk
Yeah.
because he's coming here
Oh, yeah.
to
Right,
Columbia. He's moving the Diamond here to Columbia.
Oh,
And I probably shouldn't announce this, right? But it was announced at the retreat.
Of course.
okay.
I think it's public knowledge.
So the micro retreat is only one day now?
Yeah, this is the first year because
Because they kept getting kicked out of hotels.
no, people just didn't stay for 2 days, and so
Oh, okay.
the chairman said last year, yelled at us. He said, "You know, you guys, I spend $40,000 on this, and you don't even show up." So
Okay.
it was one day. Anyway, David Ho told us the story of how he set up the Diamond
Yeah.
years ago. And the cool thing is, so David was a physician, went to medical school at Harvard. He was working in LA. He saw the first 3 of those 5 AIDS cases that were reported in morbidity and mortality.
Yeah.
And then the lady who gave the money, what was
Irene Diamond.
Irene Diamond said, "I want you to be the director."
Yeah.
And that's it. And then he hired you at some point, right?
He hired a group of, I believe, 4 junior investigators. I was one of them.
One of them.
And that was the start of the center, which has been very successful.
Mm-hmm.
And as you're saying, may be moving its location in the near future.
Yeah, well, he gave a big talk about how, they've negotiated for a long time and they're moving here. They're going to get 2 floors in this building. And that's why there's all kinds of construction noise going on here.
So at the Diamond, were you obliged to still apply for NIH grants, or did the Diamond support you?
No, it was a soft money position. You had to raise money from NIH grants, yeah.
Because I was at Rockefeller as a postdoc when you didn't have to do that. Everybody got paid.
Rockefeller had a very nice as a large endowment and can offer that, but most places
they changed rapidly during the Frederick Seitz era.
The Diamond endowment is much smaller, I think.
Yeah, much smaller than Rockefeller. Oh, of course, yes.
I think
most endowments are smaller than Rockefeller's. The name kind of suggests that.
I think someone told me it was $50 million or something. It's less than $100 million, which is surprising.
Really?
It kind of is, yeah.
That's why it's really soft money.
Well, the Aaron Diamond Center was, relatively small institute focused on HIV.
Yeah.
So that was kind of an innovative, a new idea, was that you would have, this one institute totally focused on one virus. And that was part of the vision of Irene Diamond, that that she saw this as a big problem coming up in New York City, and she wanted to do something for the city. Her husband had, Aaron Diamond had earned a lot of money as a real estate developer in the New York area, and she wanted to do something for New York City.
You bet. Now it's moving here.
Yes.
All right.
So, Ned, you started off in immunology, and did you sort of jump or drift into HIV? And I mean, how?
Right. So when I was in the Baltimore lab, that was in the early 80s. And at the time, we were just learning about the AIDS crisis. We heard the first reports of this disease that was spreading in the gay population. And initially, its cause was a mystery. People thought maybe it had to do with the lifestyle thing. Maybe it had to do with drugs that were circulating in the gay population. And then the French group, the Montagnier group in Paris, isolated this retrovirus. We were all quite shocked that this disease was a retrovirus, because we thought retroviruses were in mice and chickens and cats, but they weren't in humans, except the only thing was there was human T-cell leukemia virus. That was the only one, but that was just affected a few people, didn't cause too much problem. And then this retrovirus pops up in humans, and then they sequenced it, and it became clear that this was a virus that was affecting the immune system. So that seemed like a great, that was going to be of great importance and something of great interest. Then it intersected with virology and immunology.
Okay.
So that was how I got into that.
This is
I don't think that doing HIV and immunology is all that strange, seeing as how that's sort of similar to how I got into it. So it makes a lot of sense to me.
Well, the disease itself is the loss of the CD4 cells. So it's an immunodeficiency. So it is very related. And, I remember when Bill Hazeltine came and gave one of the first talks about HIV at the Whitehead Institute at that time at MIT. And he'd come back from a meeting where they started to talk about the epidemiology, and he said, there's probably going to be millions of people infected with this virus, and it's starting to look like almost everybody who gets it is going to die from it. And that really hit me. I mean, I thought, this is going to change our lives. It's going to change the society. And, I've learned some immunology. Maybe this would be a good field to go into.
So are you a virologist or an immunologist?
Well, I'm really I'm a virologist, but a wannabe immunologist.
Okay.
And our group actually, when we start talking about the work, you'll see that our work is leading in the direction of immunology.
Yeah.
And
that's what impressed me looking at the papers, is that, at least the sequence of papers that I read started out in a hardcore virology, and then the most recent, your hardcore immunology. But obviously, based on your history, it's been there all the time.
Yeah, because the Baltimore lab really was they were interested in B-cell immunology, but I was for a time I was in Mike Bevan's lab at MIT, was a T-cell immunologist, and so that's my roots were really immunology.
Except for a little group of poliovirologists down the hall.
That's right.
Right.
But I never really understood what they were all doing.
No, nobody. We didn't either. So you sent us a bunch of papers on a protein called SAMHD1. Let's talk about that. What is it? What does it stand for?
Yeah. So it's an HD sterile motif HD protein. I mean, people can't agree on exactly what it stands for, but it is an enzyme that falls into a class of enzymes that has this H and D amino acid motif, and they seem to be involved with nucleotide metabolism in cells.
So it's an enzyme, and what does it do?
Well,
in cells normally, without thinking about it.
Right. So first of all, we're still just understanding what it does. But one thing that it does is to takes off the 3 phosphates from trinucleotide deoxynucleotides. So triphosphate deoxynucleotides, or much easier to say DNTPs, deoxynucleotide triphosphates. It just cleaves off the 3 phosphates. Now, these DNTPs are the building blocks for DNA.
Right.
So there are 4 of them, A, C, T, and G. And when cells replicate their own chromosomal DNA, they string those nucleotides all together in order to make their genes. So it turns out that this enzyme is helping cells to regulate the concentration of DNTPs in the cell. Mm-hmm. So it's negative. It's turning down the concentration. That is taking off the triphosphates, and then they cannot be used to make DNA.
And this is a nuclear enzyme, right?
It's in the nucleus. It has a nuclear localization sequence on it.
Because not every cell in our bodies are dividing, so you don't always need to have big pools of triphosphates.
Yes,
right?
And so that is going to become important when we talk about its role in virus replication.
It has no effect on NTPs, right?
No.
RNA.
It does not affect ribonucleotides.
Is there a similar enzyme for RNA triphosphates? Do you know?
Not sure about how it works for RNA.
Should be, right?
I don't think so.
Yeah.
But not that I know of.
All right. Now,
cells that aren't replicating would still be transcribing.
Yeah.
Exactly.
That's correct. Yeah.
So that's going to be going on a lot more than.
Yeah, maybe. We're being logical here, but cells are not always logical.
Cells are not always logical. There could very well be an enzyme that does.
So you didn't discover SAMHD1.
No,
right?
we did not.
Right.
SAMHD1 had been known for a long time, but it was not known to have any role in retroviral replication. Okay. That was discovered by 2 groups. One was Monsef Benkiran's group in Montpellier, France, and Jacek Skowronski now in Ohio. Sorry. And so the way that this came about was they were trying to understand the role of one of the lentiviral accessory proteins called VPX. So just to give a little bit of background here before we jump into this, when we many years ago when we first saw the sequence of HIV, it was remarkable in that it had these extra little open reading frames. So in other words, little genes in the virus, which had not been seen in any other retroviruses. So the retroviruses like murine leukemia virus or the avian retroviruses, they only have 3 genes. So HIV had those 3 genes. And then it's got these other little open reading frames, possibly genes or possibly nothing, in there, and that were apparent from the first sequencing of the virus. So it quickly became clear that those little open reading frames do make proteins. Mm-hmm. They are genes. And so they came to be called the regulatory or accessory genes of the virus. And they all got little 3-letter names, as virologists like to call genes. So, and the names were given on the basis of what was thought to be the function. Mm-hmm. And the way you do the function is you knock out the gene in the virus and you see how it affects virus replication. So, for example, those genes are Rev and TAT. Those were perhaps two of the first ones that were understood, because if you knock them out, the virus cannot replicate. So Rev was the regulator of virion proteins, and TAT was the transcriptional activator protein. Then there's VPR, VPX, VPU, and NEF. Now, NEF is kind of a funny one because that stands for negative effect factor. Now it turns out that the role of NEF is it's a positive effect factor. Okay. So it was given that because of very early experiments, people were doing it in a big hurry and thought it was a negative factor. It's not. It's a positive factor. But, once you got the name there, it's stuck.
Lot.
So,
yeah.
Right. So now these, the TAT and REV, those are really essential proteins. These other genes, if you knock them out from the virus still can replicate pretty well, at least in some types of cells. So that makes it much harder to understand what they do and something of a mystery, because clearly the virus doesn't go around carrying genes that it doesn't need. It's very economical. So it turns out many years later, now that we know the function of these genes, more or less, to some extent, some better than others, that the major function of these genes is to counteract antiviral systems that cells have. Those antiviral systems, they are kind of part of the immune system, but they're not part of the standard, what we call the adaptive immune system. They're part of the innate immune system.
Right.
The innate immune system is this kind of collection of antiviral systems that cells have developed. And the innate immune system is not so antigen specific. It's specific more for various classes of pathogens. And it doesn't have the kind of a memory. It's not like B cells or T cells that have a receptor, antigen specific receptor, but they work to fight against certain characteristics that viruses have. And so, perhaps the best example of that is the VIF gene of the virus. So that stood for virion infectivity factor. For many, many years, nobody could figure out what it does. But Mike Malam's group identified the target of VIF and showed that what VIF was doing was binding to a host protein called ApoBec, or ApoBec3. ApoBec3 is a very powerful antiviral protein that is expressed in T cells. It's an enzyme. It mutates viral DNA. And in mutating it, it basically, as you might technically speaking, would say, it screws it up. So,
and I heard you years ago call it a WMD.
All right, that's weapon of mass deamination, right?
Love it.
That sounds good.
That was in the Saddam Hussein.
That's
era.
right.
But so it deaminates cytosines in DNA, thereby converting them to uracils and causing a mutation of the DNA. So that's just one example of where an accessory protein of the virus is counteracting a specific antiviral protein. And what I think is so remarkable about this whole thing is that the viruses have really taught us about things in cells that we didn't know about. That is, by studying the virus, that's how you learned that there was this antiviral system, because you knock it out in the virus. Now the virus can replicate. Now you've detected an antiviral system that you didn't know about. So that was how that was detected in the first place. And that continued to happen as these accessory genes were discovered. And so the host factors are called restriction factors. The accessory proteins counteract the restriction factors. So ApoBec is a restriction factor. So when these the Benkiran group and the Skowronski groups were trying to understand what VPX did, the idea was it's probably counteracting a restriction factor. So they did mass spec analysis to figure out what protein in the cell VIF interacted with, and came up with this enzyme SAMHD1. So that was how that was discovered.
Now, VPX is in HIV-2 genome, right?
Right. So that's also an interesting question in this field. So HIV2 and SIV, so, and the SIVs and HIV2 are quite closely related.
Mm-hmm.
They encode VPX. HIV1 does not encode a VPX. And so that's one of the questions that's still outstanding in the field. Why does HIV2 or SIV need a protein that HIV1 does not have?
So even the chimpanzee ancestor of HIV1, SIV-CPZ, that does not encode VPX?
No VPX.
Wow.
So what's the difference exactly between VPX and VPR?
Right. So it turns out that another accessory protein is VPR, and those two proteins are quite similar to one another in amino acid sequence. I forget exactly, but maybe I think around 50% identical. I'm not sure. I don't want to say the number. I'm not sure exactly how similar, but it's clear that they arose from a gene duplication. So one idea was that maybe they have very similar functions, VPR and VPX. However, that's, at least for SAM-HD1, that is not the case because VPR does not interact with SAM-HD1. At least HIV1 VPR does not interact with SAM-HD1. So that leaves open still the question of what the function of VPR really is. Although I think, as we'll get to a little bit later, I think we're starting maybe to get some clues about what VPR does. But anyway,
now so VPX is in HIV2. Is VPR in both viruses or only HIV2?
Yeah, so all the viruses have VPR.
Have VPR. Only HIV2 has VPX and SIV.
Correct. Yeah.
Okay.
Some SIVs, right?
Some SIVs, right?
So I guess the old world SIVs, right?
Well, there's one SIV that does not have, that only has the VPR, and that's African green monkey SIV only has the VPR, but no VPX.
An SIV.
However, if you look in that VPR from the SIV AGM, it binds to SAM-HD1.
Okay. All right.
Okay.
I think that's why I was confused about their similarity.
Right. It is confusing. And these two proteins, I think they're kind of one of the last mysteries in the molecular biology of the virus, as being like genes that where we don't really know exactly what they do. But I think that recently there's a lot of clues emerging about how they work.
So you sent us a Nature paper on which you're an author. It's called SAM-HD1 restricts the replication of HIV-1 by depleting the intracellular pool of deoxynucleoside triphosphate. So give us a kind of overview of what you did to reach that.
Right. So, after SAM-HD1 was identified as the target of VPX, we started trying to figure out, well, how does it restrict virus replication? What we knew is that the block to virus replication was at reverse transcription. Mm. So if you, for example, take out VPX from SIV, then there's a block at reverse transcription. Now, the SAM-HD1, the enzyme, fell into this class of enzymes that looked like it might have something to do with nucleotides. And the Goldstone group then showed its enzymatic activity. They showed that it was a triphosphohydrolase. That is, it could take off the 3 phosphates from the DNTPs. So we said, well, I mean, maybe the way this works is that the SAM-HD1 just takes off all the phosphates from the DNTPs, and that would cause a block to reverse transcription because you wouldn't have the building blocks to make viral DNA. So what we did in that paper was to make a cell line where we transfected it with SAM-HD1 expression vector so that the cells would now make the enzyme. And then when we infected the cells with SIV, we showed that there was a block to infection of the cells. And then we measured the amount of DNTPs in the cell. And we showed that essentially the DNTPs were gone. Mm-hmm. It was a really pronounced effect that you just kind of lost almost all of the DNTPs in the cell.
So that means cytoplasm and nucleus, right?
I think they can circulate from nucleus to cytoplasm. So, yeah, they were lost from the whole cell, and then that provided an explanation of what the antiviral activity of SAM-HD1 was.
So were you starting with cells that did not have SAM-HD1?
Yes.
And when you stuck it in, did it affect the growth of the cells?
Right. So that's an interesting question because, if this enzyme comes and depletes all the DNTPs, the cells are not going to be able to grow. Well, it turns out that the effect that we saw only worked when we caused the cells to differentiate into non-dividing cells.
Okay.
So this is a cell line, one of these monocyte type cell lines called U937 that you can cause it to differentiate by adding 4-bol ester. When you do that, the cells become kind of like macrophages. They stick to the plate, they differentiate, they stop dividing. And when they do that, now you can deplete the nucleotides and it doesn't hurt the cells. The cells don't need the DNTPs.
Yeah. Right.
So in a way, it does make some sense because the cell doesn't need the DNTPs and maybe it wants to get rid of them so that viruses don't come in and take advantage of them.
Now, if you deplete the DNTPs before differentiating the cells, that kills them?
Well, we found very little effect of SAM-HD1 in dividing cells. And I believe that's just because
the cells probably had something to shut it down.
Well, I don't think it's shut down. I just think they're making a lot of DNTPs.
Yeah, too much.
Okay.
Overwhelmed SAM-DH1.
Right.
HD1.
Yes.
Yeah.
And there's some idea that maybe what the cell likes to do is make the DNTPs and then after a while get rid of them just so that the DNTPs are always fresh.
Right.
Because they can get oxidized or damaged.
Right.
So it's a way of kind of circulating the DNTPs in the cell.
Mm-hmm.
So let's think about the entry. HIV is or SIV is infecting a cell. The nucleocapsid ends up in the cytoplasm, and then it starts reverse transcription. So that depends on NTPs getting into the nucleocapsid, starting in the cytoplasm. And so that's what we're blocking right there in the cytoplasm.
That's right, yeah.
Mm-hmm.
Yeah.
Okay. And then as it goes in the nucleus, the DNA is already made. And from that point, it integrates into the nucleus, right?
Well, I think that's the standard way of thinking about it. There is controversy in the field exactly where all these things happen. Some people think there's some reverse transcription even in the nucleus.
Sure.
But in general,
starts in the cytoplasm.
It certainly starts in the cytoplasm. Whether it's totally finished in the cytoplasm, not completely sure.
But SAM-HD1 is in the nucleus, right?
SAM-HD1 is certainly a nuclear enzyme. It has an NLS sequence on it, nuclear localization sequence on it. We have a paper where what we did was to take off the NLS from SAM-HD1. And when you do that, the enzyme becomes cytoplasmic. However, it's still antiviral.
Yeah, it makes sense.
So it doesn't have to be in the nucleus to be antiviral. And then that makes sense because the nucleotides can go from the cytoplasm to the nucleus. But I'll just say just one other thing, just in that paper, what we showed though, was that when you make the cytoplasmic form of SAM-HD1, so take off the NLS, now VPX no longer works. So to back up a little bit, what we didn't mention is that the way VPX is working is that it binds to SAM-HD1 and it causes SAM-HD1 to be ubiquitinated. So in other words, it gets little flags for proteasomal degradation. Right. And then that induces the rapid degradation of SAM-HD1. So what we believe is that these interactions are all happening in the nucleus of the cell. When we take SAM-HD1, put it in the cytoplasm, now the VPX can't work anymore.
It's not degraded.
No, it's not.
So there's a nuclear degradation system?
Yes. So we believe that the specific proteasome, it's proteasome, and then these factors that called Cullen factors that they associate with VPX and SAM-HT1 in the nucleus of the cell, and then the enzyme gets degraded in the nucleus. So the nucleus has all these proteins that are cell cycle regulated.
Yeah.
And those are getting degraded in the nucleus, I believe.
Right. Of course.
Right. Now,
So
in a normal cell where SAM-HT1 is in the nucleus, the virus is going to come in and it's going to try to reverse transcribe or start its reverse transcription in the cytoplasm. SAM-HT1 is chewing up the DNTPs in the nucleus, but that also depletes the pool in the cytoplasm?
That's the idea, yeah.
Okay.
And the other factor which we should mention is that this protein VPX is present in the virion itself. So that's a kind of a distinguishing feature of VPX and VPR is that they are in the virus. So the other regulatory and accessory proteins, they are active in the cell, but they are not in the virus particle, like TAD and REV. They're not part of the virus. So the fact that these things are in the virus was kind of a clue as to what they would be doing, that the virus wants to bring them into the cell when it starts the infection.
Where are they in the particle?
Well, that it's not totally clear where they are. It's not that easy to figure out. I mean, we thought that they would be inside the capsid, or at least that VPX would be inside the capsid, but not really clear. We think that that they're probably located outside of the capsid so that they can be released very quickly into the cytoplasm of the cell.
Yeah.
So this brings up something about HIV structure that I have ruminated on for a number of years. When if I understand HIV assembly correctly, it starts out with a spherical particle that looks fairly homogeneous in the electron microscope, and then basically late during the assembly, the core forms inside, and that's sort of a gumdrop shaped thing inside this sphere, and there's room between the core, the outer core wall, and the undersurface of the membrane surrounding it that, for me, in my mind, has always looked like the equivalent of like the herpes tegument or the vaccinia lateral bodies that classically carry proteins like this that are sort of the advanced guard on infection. The particle gets in and it's got these proteins ready to go and go out and trash a bunch of cellular things that are trying to fight off the infection.
Yes, no, I think that's the right idea. That's probably where these proteins are located, so that they can be released quickly when the virus enters the cell.
You have go ahead.
The amusing thing to me is that the bit about if you relocalize SAMDH to the cytoplasm, VPX doesn't work. That's probably because VPX is going to the nucleus looking for SAM-HD1, and it's not there, right?
Yes. So if you do immunofluorescence and to look where VPX is in a cell, it is a nuclear protein.
But if you took the NLS off of VPX, then it should work on a cytoplasmic.
That's correct. However, that experiment is hard to do because VPX does not have a clear NLS that you can take off.
Right.
I think it's exactly how VPX is in the nucleus. We don't really know.
You have a paper which we also saw. Degradation of SAM-HD1 by VPX is independent of uncoding, so that relates to where it is. It's probably not in the capsid, but that leaves, as Rich said, between the capsid and the membrane, somewhere in there.
That's what we were getting at.
Can't just take a picture of it?
Well, you would think with an electron microscope you can, and people have tried to do that. But the pictures were not that clear. It's not that easy. Right. You need these gold particles.
Yeah, how about Jason Mercer's techniques? I forget what they were called, but the high-resolution microscopy techniques. This may be an opportunity to employ them to get some
talk about that on recent TWiV.
Yeah. So they looked at the distribution of the proteins on the surface of pox viruses, and they could see that they were skewed. Attachment were on the sides, and fusion proteins were at the end. And this is some kind of high-resolution microscopy coupled with computational interpretation. Maybe.
It'd be worth doing something like that.
Or a micro-CAT scanner. Right?
What?
We just did a paper on TWiP about ants that are infected with parasites, and they put them in a micro CAT scanner, and they could see the parasites inside the bloody ants.
This is true.
Really?
Wow.
Down to 1 micron resolution.
That's cool.
And it was clamped onto its main ganglion, and that's how it controls its behavior.
Wow.
It's so creepy. The thing is
God.
full of them. Anyway, the other thing is you
you have
also
to tell the ant to hold very still, though.
well they were
They were under the influence of alcohol
they
at
were,
the
they were
time.
fixed. They were fixed. Yeah, Dickson and I were talking about that because he wants to do it with Trichinella infected mice,
That's right.
but the mice are going to be moving. You know,
They could be anesthetized.
you could say stay still.
They could anesthetize.
People do that on mice.
Stay still for half an hour. Ned, you also looked at other retroviruses to see if they're... So HIV-1 is susceptible to SAM-HD1 and HIV-2. What other retroviruses are susceptible?
Well, I think that they are all susceptible because they all need nucleotides to replicate.
Right.
But you said foamy viruses were not.
All right, that's a good point. Yeah, except for the foamy viruses because they make the DNA before the virus.
Before they leave the cell.
Thank you for reminding me of that.
So the next jump from this
Right.
study is an animal model.
Wait. We're not done.
All right.
Before you get to animals. So, outside of retroviruses, have you looked at any other other DNA
Yes,
viruses?
herpes viruses are. We didn't look at that, but other people did look at it and showed that they were sensitive to SAM-HD1. Yeah.
Yeah, that makes sense because they
Yes.
have to. Now, Rich, what would you think for pox? Do they have their own DNTP synthesizing machinery, or maybe it wouldn't matter?
They make some nucleotide metabolism enzymes, but I would, this might have an effect.
It should, because a triphosphate is going to be hydrolyzed.
Well, first of all, these other viruses may have other ways to deal with the SAM-HD1 problem.
Yeah.
There is an enzyme encoded by herpes viruses called ribonucleotide reductase.
Right.
And that is an enzyme that converts ribonucleotides into deoxys. Okay, so in a way, that's providing a source of DNTPs, and that's kind of counteracting.
Yeah, and pox viruses have the same thing.
And that enzyme probably made the RNA world into a DNA world in part, right? You had to make DNTPs. That's one of the ideas that ribonucleotide reductase.
Are there any viral oncogenes that are known to interact with SAM-HD1?
Viral oncogenes? No, not that I know of.
Okay, because I'm just thinking that a side effect of turning off SAM-HD1 would be that you now have these DNTPs floating around. So if the cell was going to replicate,
yes, that's right. Could have. There is some indication of roles of SAM-HD1 in cancer, and that may have to do with DNTPs. It's also worth pointing out about SAM-HD1 that this is really the restriction activity or the antiviral activity of SAM-HD1 really only occurs in certain types of cells.
Right.
So in actively replicating CD4 T cells, which are the major target for HIV, SAM-HD1 is not antiviral. It doesn't have any viral activity, and there's lots of DNTPs present. This feature is really something that's present in the myeloid cells. So those are like macrophages and dendritic cells.
Dendritic cells. Yeah.
So those are important targets for lentiviruses. They do express CD4 and the co-receptor. And so SAM-HD1 is important in these myeloid cells, but not in actively replicating T cells.
So is that
those
why
myeloid cells, like DCs, they can become reservoirs for HIV infection, right?
Yes. So, they can, because when HIV infects a macrophage, it tends not to kill the cell, unlike actively replicating CD4 cells are killed pretty quickly by HIV. Macrophages, they get infected and then they produce virus, but they're not really killed by the virus. So they can become reservoirs to some extent. But, the HIV does not infect them that well. It does infect them, but, the SAM-HD1 is blocking macrophage infection somewhat. What it's really blocking very tightly is dendritic cells. And so I think that is something that we're going to get to. But dendritic cells express quite a bit of SAM-HD1, and their DNTP levels are very low. And so they're blocked from HIV-1, but they're not blocked from SIV infection.
Because of VPX, right?
Yeah.
So
or how about HIV-2?
It's HIV-2 as well. Yeah.
HIV-2.
Okay.
Are there any polymorphisms in humans in SAMHD1 that influence HIV susceptibility or prognosis?
Right.
Question.
A very good question. In fact, there are. So there's a disease, which is a rare neurological syndrome called Aicardi-Goutieres syndrome, AGS for short. That syndrome is, it's a childhood disease, severe neurological problems. It appears to be the result of excessive interferon production. And in a way, it's kind of like, almost looks like a chronic virus infection. So there's, children make a lot of interferon. It causes neurological problems. That disease can be caused by any of 5 different genes. The classic one is called TREX1. But it turns out that deletion of or mutations in SAMHD1 cause AGS as well.
And MDA5 and ADAR.
Right.
And ADAR.
And also ribonuclease as well.
We just hired someone from Charlie Rice's lab who works on ADAR.
Oh, okay.
And she's going to work on that. That's why. So ADAR deaminates double-stranded RNAs so they don't stimulate the innate response.
Yeah.
Right.
Right. Yep.
And that's why SNPs and ADAR gene are associated with this disease. I don't understand why SAMHD1 SNPs would be in this.
So that's getting into a whole very new and confusing but extremely interesting area of just SAMHD1. So it turns out that SAMHD1 appears to have other functions in addition to depleting DNTPs. It appears to have a role in the repair of damaged DNA.
Yeah.
And if damaged DNA is not repaired, it results in little pieces of DNA going from the nucleus into the cytoplasm. Now the cytoplasm has a detector for cytoplasmic DNA
CGAS
called CGAS.
Yep.
And CGAS, it's kind of looking out for cytoplasmic DNA because cells are not supposed to have DNA in the cytoplasm.
Yeah.
And so it may be that all of these gene knockouts are things that result in cytoplasmic DNA. So TREX1, for example, is an exonuclease. It degrades DNA. So it is also maybe getting rid of cytoplasmic DNA. When you lose it, now you got cytoplasmic DNA, you got these innate immune response. So you get this just chronic inflammatory responses that causes.
So it can be RNA
diseases.
or DNA that can lead to this, because MDA5 is an RNA sensor, right?
Yeah.
ADAR is an RNA deaminase and so forth. So that's really cool.
And that's the paper that came out earlier this year, correct? The replication paper with SAMHD1?
Yes, that's correct. That was a paper earlier this year. The initial paper just looking, there was a paper several years ago from a German group where they just showed that if you got the T cells from a patient with AGS who has a SAMHD1 knockout, then now you can infect their cells better. Their macrophages are more infectable with HIV.
Okay.
So is the lack of VPX and HIV-1, is that made understandable by the low amounts of SAMHD1 in the primary target, CD4 T cells?
Yeah, but that doesn't quite explain the difference between humans and non-human primates. In other words, the T cells of humans and monkeys both have lots of DNTPs.
Yeah.
But it seems that, at least what I think makes sense, is that in HIV, the infection is very much the virus is infecting T cells. Those T cells then make more virus, and then other T cells get infected. Maybe in SIV, the infection is not quite so simple. Macrophages are getting infected, or dendritic cells getting infected. Then they pass that virus on to T cells. So it's not so much a T cell to T cell infection, but rather a macrophage to T cell infection. And that's why SIV would need VPX in order to be able to infect this type of cells. So we know that T cells and macrophages or dendritic cells, that they interact during an immune response. That's right. Their job. They collaborate, they cooperate together. And so it's a very efficient way for a virus to spread. It infects the macrophage. When the T cell comes along, now the T cell gets activated by the macrophage, and at the same time, the virus gets handed off.
So is the SIV CPZ more like HIV in the T cell to T cell spread because it lacks VPX, you said?
Well, that would be the theory. But I mean, I don't know that much about SIV CPZ, but that would be the idea.
This could also maybe explain part of why HIV-2 has a very different pathogenic process than HIV-1, right?
That has also been suggested, yeah. Well, it's been suggested that the ability of the virus to infect dendritic cells results in a better immune response against the virus.
Right.
And maybe that's why HIV-2 isn't quite as pathogenic. I mean, this is speculation now, but
sure,
that is possible.
So maybe we could move into now using VPX. So you had a paper where you put it in a lentiviral vector and did some interesting things. So maybe you could tell us the thinking behind that.
Yeah. So as we've been saying, HIV-1 doesn't have a VPX. So we kind of wondered, why it doesn't have it and what would happen if we provided it. Now, it's not that easy to do, but we engineered an HIV-1 that now could package VPX proteins into the virion itself.
So it was a gain of function experiment.
It was a gain of function experiment. Yes.
Except you didn't clone the VPX into the virus. You had to supply that from another vector, right?
That's correct. That's how we did it. We actually did one experiment. We did clone it into the virus, but it kind of messed up the virus. Didn't work that well. But yes, we provided in trans, as they say. So what we did was we figured out how VPX gets into the virion in the first place. It gets into and there was some studies already known about this, that the way it gets into the virus is it binds to the major structural protein of the virus, the GAG protein. It binds to the portion of GAG called P6. And there's a little amino acid motif that we defined there in P6. And that's the thing that VPX binds onto. HIV-1 is lacking that sequence. We transferred that little sequence into HIV-1. And so now when we put that viral DNA into a cell, and then we also provide an expression vector that makes the VPX proteins, viruses get made, and those viruses contain high copy number of VPX.
Mm-hmm.
Now you take those viruses, and now you try to infect dendritic cells, and now all of a sudden it works. So HIV-1 could not infect the dendritic cell. Now we put the VPX in there, and it does.
Because it's antagonizing SAMHD1.
Because it's antagonizing SAMHD1. So, you might have said, well, HIV doesn't have VPX because it doesn't need it. It has some other way to infect dendritic cells. It turns out not to be the case. It's totally dependent on VPX. So when we provided that, now we could infect dendritic cells. And that seemed to have some potential therapeutic implications because people use these lentiviral vectors, which are vectors based on the HIV-1 genome, and they use them to express whatever protein they want in a cell. These lentiviral vectors don't work so good in dendritic cells because SAMHD1 blocks them from working. So now we had a way that we could transduce dendritic cells. That is, we could use lentiviral vectors in dendritic cells.
Or any myeloid cell. Right?
Or, any myeloid cell. But dendritic cells are particularly of interest because, they're really good. They're really important in the immune system. So dendritic cells were identified by Ralph Steinman when he was postdoc in the Cone group in Rockefeller University. And they were trying to understand how antigens are present. Right. This is about the early about 1973 that paper came out. So they were trying to understand how antigens got presented to T cells.
Yeah.
And they identified this kind of strange looking, very rare population of cells that had little projections. They called them dendritic cells. And they said, oh, the antigens are getting presented on these cells. And people initially didn't really believe it, but it turned out it was quite true. And these cells are really very important in initiating immune responses. And, as you know, in 2011, Steinman got the Nobel Prize for the identification of these cells. And people came up with the idea that, well, we could use these cells as a vaccine. That is, we could put antigens on them,
right,
and then inject them into a person, and now we'll get an immune response. For example, a cancer is known to be, in some types of cancer, can be controlled by an immune response. So if you could stimulate that anti-cancer immune response using dendritic cells, it might be a cancer vaccine.
And specifically, you would want to induce a cytotoxic response.
Yeah, so the CD8 positive CTL cytotoxic T lymphocytes would be the best way because they will kill the tumor cells.
Right.
And so this idea of dendritic vaccines has been something that people have been working on for a long time.
How could you make a dendritic cell favor CD8s rather than antibody production, say?
Right. So, these are all the questions that people are working on in this dendritic cell vaccine field. But the CD8 T cells are T cells that recognize little bits of antigens, that is peptides, small 8 or 9 amino acid long peptides. Those peptides are presented on MHC, major histocompatibility protein complex type 1 proteins.
Right.
So the peptides are present on the surface together with these MHC proteins. And the CD8 T cells have a T cell receptor that sees that complex. So if the dendritic cell is presenting the antigen together with MHC class 1, that stimulates the CD8 T cells.
Whereas if it presents it with class 2, that stimulates the CD4 and the helper response and the antibodies.
That's right. Yeah. So the idea was you could take dendritic cells and then maybe just incubate them with synthetic peptide. That peptide could have like a little epitope or little piece of a virus on it. Inject those cells into a person. That would stimulate the the CD8 T cell response, and now you could get a response against a tumor or against a virus. And it has been successful to some extent. So there is, in fact, a licensed vaccine, which is a prostate cancer vaccine called Provenge, that it works exactly that way. And it provides some, several months of additional
So you have to take
life.
DCs out of the patient?
That's correct.
And add the vaccine and put them back in. Right?
The way they do is take the DCs out or take some blood cells out, and then you got to culture them with a cocktail of cytokines to cause usually you start with monocytes.
Nice.
So these are the precursors for dendritic cells. Differentiate them with cytokines into dendritic cells. And then in addition, you have to do something called mature the DCs. So that is, the DCs have to become like good at antigen presentation. They got to be able to home to lymph nodes. They got to be able to make cytokines that stimulate T cells. And so you do all that stuff. It's a whole bunch of cocktails of cytokines. Add your peptide or whatever is your tumor associated antigen or whatever, and inject them back into the person. And so
this is not cheap or easy.
No, right. No. Very expensive and difficult, and it hasn't been that successful. But what we thought was, well, maybe now that we can, with our fancy lentiviral vectors, now we could actually express the antigens in the dendritic cells rather than just put them on the surface.
Right.
And there's a lot of advantages to doing that, because when the cell makes the protein endogenously, the protein gets assembled together with the MHC protein very efficiently. Much better than when you just add it from the outside. And plus the cell will just keep making the antigen, rather than just that one-time addition.
Yeah. So when in the course of the SAM-HD1 story did this light up in your head that I could use this to put them in DCs? and do this that you just explained to us?
Well, I think that was when we saw that how nicely that worked. We were thinking, well, what good is this?
Yeah,
So it wasn't that difficult to think about.
I think it's a brilliant illustration of how you do some fundamental virology. What is this VPX doing? We have no idea. We have no therapeutic end in mind. And we find it interacts with SAMHD1, and then it ends up making a vector that can go into myeloids. I just love that kind of progression. Right?
So, but it is related to what we were talking about a few minutes ago, and that is that the idea of HIV not infecting dendritic cells and therefore not making as strong an immune response.
Sure.
So we figured, well, we'll just kind of override that. We'll get now the virus proteins made in dendritic cells. So we're kind of overcoming that that obstacle, and maybe now we get a better immune response. And so that is exactly what we've been doing, working on quite hard, and hope to publish that data soon. And that is showing that we could make an anti-HIV vaccine using this approach.
So you published a gene therapy paper showing what you could do. You could deliver epitopes this way. Tell us about what that is.
Right. So that was kind of the precursor to trying to make an HIV vaccine. So, and this is a therapeutic vaccine. So it's not the type of vaccine that you think of for polio where you prevent the infection. It's a vaccine that will be given to somebody, presumably somebody whose virus loads are suppressed using antiretroviral therapy, with the idea of strengthening their immune response against the virus so they wouldn't have to take so much drugs. So, what were you asking me?
So, tell us what you did in the gene therapy paper.
So the idea is, could we use this as an HIV vaccine? So what we did was we took our lentiviral vector system and we cloned into it just one of the most well-known CTL epitopes for HIV. It happens to be called SL9. It's a part of the matrix protein of the virus. It's just a short amino acid sequence. So we clone that into the vector. And then at the same time, we cloned in a gene called CD40 ligand. Now, CD40 ligand is a protein that's expressed on T cells, and its job is to cause dendritic cells to become activated and mature. So as I was saying, dendritic cells work much better when they're mature. So we figured, well, we'll express the antigen and we'll express a gene that activates the dendritic cells. And now we'll have a nice dendritic cell vaccine that'll work better than the earlier ones. And so Tom Norton, who's actually a clinician scientist in the lab, did most of this work and continues to do most of this work in our lab. He then tested to see whether that worked. And the way he did it was we took a CTL clone. So this is a clone that a human cell line. It's a cytolytic T lymphocyte. It has a T cell receptor that sees the SL9 peptide on the context of MHC class 1. So he took his dendritic cells, infected them with that lentiviral vector, then incubated with the CTLs and asked, did the CTLs recognize that complex? And they did. The CTLs got stimulated very strongly. So we knew that the antigen was being presented. We could also show that that you could activate T cells that way. T cells would get primary T cells would get activated as well, just from a human. So we knew then that this was working in vitro. We also had this idea that maybe we could use it as a way to kind of get at this very stubborn reservoir of latently infected T cells. So that's been a big topic of research. That is, why can't you cure somebody of HIV? And the reason you can't cure them is because they have just a very small number of T cells that harbor a latent virus. They harbor an HIV that's silent. But every once in a while, that T cell gets turned on and it starts producing virus. The current drugs, antiretroviral drugs, cannot get rid of those T cells. So as a result, you can't cure somebody. Many people and huge consortiums have now formed of scientists trying to figure out how to get rid of those latently infected cells. So what we thought was maybe what we can do is have dendritic cells activate those T cells. Then they start to make virus, and then we stimulate a T cell response against the virus. So in other words, we activate the T cell and then kill it. So that approach has been termed shock and kill. A lot of groups are trying to do it. Whether it can really work or not, I don't know.
So when you activate, so did you show that you can use your DCs infected with your lenti producing the peptide and CD40 ligand? Can that activate a latently infected, HIV-infected T cell?
Yeah, so we showed that, it's very difficult to really show it with real latently infected T cells. But what we used is a cell line, which is a model latently infected cell line. That's called JLAT. Those are cells that a lot of people look at. They've got a latent HIV in there. And when we incubated those JLAT cells with our dendritic cells that had the vector in them, now they got they started making virus.
So do those T cells then die?
Some of them do. Yes.
So virus is cytopathic, right?
Oh, the virus is cytopathic. Yeah. So, one idea is that the virus, this virus, the cells would just die from the virus production.
Yeah.
And that was certainly the initial hope of how this would all work. But apparently that is not going to work because in real life, when a primary T cell gets activated, it often is not sufficient to kill the cell.
I see. So how do you end up killing that latently infected? I mean, you turn it on, it makes virus, but what do you do?
Well, in our case, the idea was that the dendritic cell vaccine is stimulating CTLs that will kill the cell. That's the idea.
Because that latently infected cell is now visible to the CTLs.
That's the idea.
Yeah, got it. Because it has peptides on the surface.
Right. So, the field as a whole is trying to develop mechanisms to turn on those latent viruses. But it has to be a two-step thing. One is, as they say, the shock. That's going to be something that causes the latent cell to make virus. And the next thing is going to be the kill. You have to have strategy to do both.
Sure. And your cytokines that are being made by that DC are probably going to help out with that shock. Is that right?
Yes. Yeah, that's a good point. So we did look to see how the DCs were turning on those latently infected cells. And what we were able to show was it was largely the result of the production of tumor necrosis factor alpha, TNF alpha, that was stimulated by the CD40 ligand in the vector. And so the DCs were making TNF, and that's then turning on the latent virus.
So what is the next step you need to do now to test the feasibility? And maybe that's what Dickson was asking an hour ago about a mouse model. I wanted to get to this point.
I did remember that question, actually.
So those are just some tissue culture experiments. How could you get an idea of whether this is going to work or not? Yeah. So there are humanized mouse models. So we collaborate with a group at the UCLA Center for AIDS Research. It's run by Scott Kitchen. And they are very good with these humanized mouse models. These mouse models are mice that have a human immune system. The way that they have a human immune system is that they're reconstituted with human bone marrow, liver, and thymus. These are fetal tissues.
Yeah.
And they will, to some extent, reconstitute the immune system. You can get production, you get T cell maturation, you got the dendritic cells, myeloid cells.
And these are mice that lack their own immune systems, right?
Yes. Yeah,
whatever.
They're immunodeficient mice, so their immune system is dependent on the human immune system, and you can infect those mice with HIV.
Right.
And these cost about how much each to make? Like $1,000?
Well, who's counting?
Right. They are expensive to make, and you got to get the human tissues. And actually, this became a hot topic in the news several months ago because of the right-wing groups who were trying to shut down Planned Parenthood.
Right.
Yeah.
So they were arguing, and actually that discussion is still going on.
That's right.
That discussion is still going on. The use of fetal tissues in research is being revived again. But
couldn't you use induced stem cells to make all the cells that you need?
there may be better other ways to do it, but they have not really worked so far. Anyway, so those mice get infected with HIV. So the experiments that are in progress now are to use the dendritic cell vaccine in the humanized mouse model. And what we can show is that you do get a suppression of virus load when you do this vaccination. Virus loads go down about 100-fold in the mouse. Although after several weeks, virus loads come back up again.
Do you get a latent reservoir in these humanized mice?
Yes, you do.
Okay.
So, I mean, you can do experiments to look to see whether the vaccine will have an effect on the latent reservoir or not. Those are doable experiments, yeah.
But why does the virus titer? So you're looking at primary infected mice that you inhibit initially, but then why does it go back up again?
Right. So that was the question: is why doesn't the vaccine keep working?
Yeah.
And that's something we're working on right now. But what we believe it has to do with is what's called T-cell exhaustion. So, I mean, the first idea was that the virus had just mutated and escaped from the vaccine. Right. However, we sequenced the viruses in the mice and they did not appear to be mutated. The next thing we did was we looked into this question of T-cell exhaustion. This is a very hot topic these days because of cancer. So it turns out that in cancer, that there is an immune response against the tumor, but the T cells become exhausted. They stop fighting against the tumor, but you can revive them, at least to some extent, make them start to be functional again by injecting antibodies against a series of proteins that are called checkpoint inhibitors.
Right.
Or checkpoint activators. So these are molecules such as PD-1, PDL1, CTLA-4. In treatment of melanoma patients, these antibodies are extremely really remarkable.
That's what happened to Jimmy Carter, right?
He got such a treatment. He's one of the people, and so what those antibodies are doing is reviving the immune response.
Yeah.
So the question is, could we do something like that with this vaccine? And that's exactly what we're doing. We cloned into the vector a checkpoint inhibitor. And now we're seeing whether that now will like get an extended viral suppression of those vectors.
And this is being done in humanized mice, right?
Humanized mice, yeah.
Now, you're targeting HIV with this, which is a great target to go after. But this problem of getting a good cytotoxic response and viruses that are chronic, I mean, this has been a problem in vaccinology and virology for a very long time. Are you looking at targeting other viruses with this approach as well?
We have not been looking at targeting other viruses, but what we have been looking at is targeting tumors.
Right.
So, it's the same idea. And the question is just, what are the antigens? Because tumors are largely self.
Right.
But there have been identified tumor antigens. So these are normal human proteins, but their expression on tumor cells is very much increased. And they can be used as antigens to kill tumor cells.
Right.
It's great. I love it. Absolutely. Hey, Rich, are you there?
I'm here.
I
love it too.
I just wanted to make sure that you didn't get cut off. And you've been screaming for the last half hour.
Well, the deal is, Ned, you are very, very clear. And so I'm just riveted to this.
Okay.
I'm just, because it's all going in. I really appreciate it. I have a question, however.
Okay.
Do you think that SAM-HD1 evolved for some cellular chore like DNA metabolism, or did it and then was gained a side job as a restriction factor, or do you think it evolved as a restriction factor?
Right. So those are good questions. My impression is that SAM-HD1 did not evolve as a restriction factor; that it evolved for cellular function. And that I think the cellular functions are becoming more understood, that it seems to be a protein involved in DNA repair. And so it's more like, maybe the SAM-HD1 just happens to be a problem for the virus. that it didn't evolve for that purpose, but now that it's there, it causes a problem.
Right.
I mean, there are, with some of the other restriction factors, you can see that they clearly were evolved to be antiviral factors, and particularly the ApoBec proteins.
Right.
So,
I remember asking somebody about ApoBec, and there was clearly it seemed it evolved as a restriction factor.
Yeah, and you can show that because of if you look at the DNA sequence of the ApoBec genes, you can see that they are very rapidly evolving genes. And they're evolving rapidly because they have to change in order to fight against the viruses.
Yeah.
Right.
And that, you can see that
if they had a cellular function, they wouldn't be able to change so fast.
Yeah, or they would not have selective pressure on them to change so fast.
Right.
Well, they could change it if they had a cellular function as long as the domains were different enough, right? Because you can see that in a lot of proteins that happen to be virus receptors. The proteins can change to prevent virus binding, but they like transferrin receptor. It maintains its function as a transferrin receptor.
Is SAMHD1 an interferon stimulated gene?
We looked at that, and we have not seen a big effect of interferon. It's on in the absence of interferon, and that is true. Most of the ApoBec genes also are not really interferon response genes. They're just on constitutively in the T cells.
Do you anticipate eventually doing some clinical trials?
Well, I mean, the next step in our stuff would be to do some non-human primate studies, and we are planning to do those. We're trying to develop the vectors right now. But yeah, I mean, the small clinical trial would be something to do.
That's something you want to do, right? You want to get involved.
Yeah, we'd certainly. I mean, we'll see how the story evolves, but yeah, that would be the idea.
By the way, do you know how quickly after you're infected does the reservoir get established in an HIV-1 patient? Is it like 2 weeks or 3 weeks?
I think very rapidly.
Very quickly.
Yes, because if you treat people very quickly after infection with antiretrovirals, you don't cure the infection. That the reservoir is already established.
Right.
So in an animal model, like a non-human primate, and you treated before you infected with HIV, could you prevent the latency?
Oh, wait, I didn't understand the question.
So you start them off on a drug therapy to prevent infection.
Oh, okay.
And then you give them HIV and see whether or not that
pre-exposure prophylaxis.
Prep.
Prep.
If you do that, they don't get infected at all. No,
in fact, you can get PrEP. You can go downtown.
You can do that in people.
Columbia offers it to its undergraduates.
So how soon?
It does.
I mean, is it days or weeks or months? We were just asking that, of course. But I mean, you think it's days or maybe.
I mean, I don't know the exact timing, but it's quick enough that if you give somebody antiretroviral therapy, just a few days after infection, that reservoir is there.
A few days.
That's incredible.
Wow.
Yeah. Usually, don't you try to do it in hours?
Well, if you want to,
exposed in the lab.
Yeah, now needle sticks, they want it immediately, You
try to do it as fast as possible. I think, I don't want to say exactly the data, but if you wait a few days, it no longer works anymore. You have to do it within a certain number of hours post-infection.
I think at the Cold Spring Harbor thing we asked them that, and they said by 2 weeks, that's it.
And it's in the bone marrow mostly? Or?
What, the reservoir?
Yeah.
Well, I don't know exactly where the reservoir is, but it's probably a lot of it is in lymph nodes.
Yeah. Circulating too, right?
Circulating blood T cells as well. Yeah.
And John Coffin called it the holy grail of HIV-1, right? The latent reservoirs. As you said, so many people want to crack it.
Right. I mean, it is. If you're a clinician treating AIDS patients, now you can give them a cocktail of drugs that's going to take the virus levels down to undetectable levels.
Right.
And now you figure out, well, but why can't we just get rid of those last few cells?
Yeah.
I mean, what's the big deal?
It's really insidious, isn't it?
Yeah.
It's amazing. It's amazing. You couldn't have planned this. You could not have planned it. It's just amazing. I don't know any other virus that does that, right? Where you have this long-lived, silent reservoir.
Well, how about how about herpes viruses? Same idea.
Yeah, I suppose. Yeah, you got latently infected peripheral ganglia. Yeah. And no one's figured out how to deal with that.
Nope.
Right.
Nope.
Right.
But there, of
partners.
course, but there you don't have to take drugs your whole life. It's only when you have reactivation that you take some drugs if you don't want to get a cold sore, and it's not going to kill you.
Right.
Right. So here it's pretty serious. Right.
Yes.
Yeah, I guess all the herpes
clearly
virus.
the approach to herpes would be to use a pseudotype lentivirus targeted to the dendritic cells with this approach.
Well, then you would destroy the neurons.
Then you would destroy the neurons. That is
and that latent herpes virus isn't mutating the way HIV is.
No. Well, all these herpes, besides simplex, you have CMV and EB. They're all latent, but disease is intermittent, if at all.
Right.
So it's not a big deal. But
And most people, yeah.
HIV, you have to keep taking drugs.
Right.
Although,
That's right.
as someone said at the meeting, it's great that you can take drugs and live a whole life, right? This, as you said, it was a death sentence initially.
Oh, of course. I mean,
So this is
there's a
progress.
real triumph of
This is
medicine to be able to
Yeah.
what has happened in not that many years. And, with a lot of people complaining that nothing was being done about this. It's not true. I mean, it was the scientists in academic labs
Yep.
and the scientists at the pharmaceutical labs who
Cool.
did this. There's something, with all this immune response thing that's very interesting, and that is that there are some individuals who get infected with HIV and they don't get sick.
Right.
So these are the elite suppressors, they're called.
Mm-hmm.
So they can be infected for a long time. Virus loads are very low. And so the question is, how do they do that?
Right.
And why can't we make everybody else like that?
Right.
Yeah.
So,
Right.
it does look like it's the immune response that's keeping the virus down. It's a good CTL response
Right.
is important. So that's kind of the direction we were thinking in with this dendritic cell
sure.
thing, is that if you could, maybe you just have to stimulate a better immune response, and then people could become more like elite suppressors.
Yeah.
Yeah.
Well, that's an MHC1 associated phenotype, right? Elite suppressors. People with certain types of MHC1.
Yeah, it is associated more with certain haplotypes of HIV. So it does. It's MHC linked. Yeah.
This is a great story. Anything else you want to tell us before we wrap it up?
Well, it should. Thinking back, I made one error
No problem.
in the very beginning.
We've made many.
When you asked me about CD4 and CD8 cloning, I realized it was.
I was actually going to ask you about
CD8
that.
was Dan Littman. CD4 with Paul Madden.
Yeah, that's what I thought.
Okay, they were right next door, and you were.
I thought that was how it worked. Yeah.
Yeah.
And that's amazing. The two definitive T-cell surface antigens, in one lab by two neighboring people. It's amazing, right? But that was a technology that had just emerged to be able to start cloning genes.
Yeah,
right?
and
Right.
And that was amazing that the AIDS, the syndrome, was initially defined as a loss of CD4 T cells.
Right.
People wonder, well, what's going
Right.
to cause a loss of CD4 T cells? Sure enough, it's a virus that uses a CD4 as its receptor.
Yeah.
It couldn't have been simpler.
But if you ask that on an exam and your students, it's a receptor, you can say, what else could it be though? You wouldn't
Right.
want a simple answer like that, right?
Well, it turned out not to be quite that simple because there's a co-receptor.
Yes,
Right.
that's right.
So, and that was what my lab was, when we were at the Aaron Diamond Center, that was our focus. So we actually, in collaboration with Dan, we had the idea that that CD4 wasn't the only thing,
yeah.
that there was going to be something else that was required. Because when we put CD4 into a mouse cell, for example, human CD4 into a mouse cell, you couldn't infect the cells.
Yeah.
So you said, there's something missing in the mouse cells. So, Ed Berger had identified what he called fusin.
Yeah.
It was an additional protein required. And so we figured, well, there's got to be another one. So we identified, working with Dan, we identified CCR5.
Right.
And just a few weeks after we identified CCR5 as being a critical factor to allow HIV entry, we had Rick Kaup, who's a clinician scientist working across the hall, and he had these patients who he said they've got exposed to HIV a lot, and they never got infected. So he gave us some of the cells. We sequenced the CCR5 gene, and there's a 32 base pair homozygous deletion in CCR5.
Yep.
I mean, that was
That's great.
like when that happened, we were just knocked over. We could not believe it. I mean,
Yeah, I remember that.
it was so quick from one thing to the next, and that it turned out that these people really were resistant to HIV infection. And that turned out to be a very important finding because it told us something about how the virus works, but it also told you about a really good drug target.
Mm-hmm.
And so now the drug called Maraviroc is a protein that binds to CCR5 and blocks infection.
So if CCR5 is missing that much of itself, that means it doesn't actually work very well in that person?
It doesn't work at all because
So what's the
32
effect
base
of that?
because 32 is not a multiple of 3.
Protein is truncated.
So then what effect does this have on the biology of that person?
They're fine. They may be more susceptible to West Nile virus.
That's
right. Do you know what? So about 10% of the human population in a certain area has a Delta 32, right?
Not quite. It is the highest concentration of those homozygous people is in like northern Scandinavia. I don't think it's quite as high as 10%. Human, like United States, Caucasian population, about 1%.
So there have been various theories to trying to explain why that was selected for in certain people. One was smallpox receptor, which doesn't seem to be the case. Another one is the plague.
Right.
Do you have any thoughts?
I have no idea other than that. Those two things. That's not it. No.
Yeah,
no, I don't know why.
I'd heard something about genetic drift and Vikings. It seemed to make some sense.
Drifting?
Certainly much more than the
They
plague
didn't drift,
did.
they rode.
That's right. Exactly right.
Cool.
I'm actually, that's actually my lecture topic for Monday. So you're all really helping me prep for next week.
Wonderful. All right. Let's move. The last thing we do on TWiV is picks. Ned, each of us pick something we like, and if you have something you've read or seen lately, you can do that. But if not, it's okay. But we'll start with Brianne. What do you have for us this week?
Well, I have a somewhat self-serving pick. So I have always really appreciated Vincent's online virology course, both in helping my students and in myself figuring out how to set up a virology course. And so I decided that I wanted to start recording my immunology course and making a parallel site. So I am actually currently recording my immunology course. And I finally have the website up and going for this current semester's immunology course. In fact, today we talked about the Tonegawa experiments and the mini-gene hypothesis that was mentioned earlier. That was actually my topic today. So hopefully this can help people brush up on some of their immunology knowledge. I hope it helps somebody out somewhere.
Cool.
I like it.
Great.
Look at this.
Nice.
You got your YouTube there. Very good. And you've got a link to your TWiV appearances, which is great.
Yeah.
Very nice.
Yeah,
Yes,
the site is nice.
Thank you. Fortunately, Vincent was able to give me some advice so that I was able to figure out how to do all of this.
This is great.
I want to transfer to your university to do this South Africa trip. This looks like a blast.
It is a lot of fun. This will be my third year and the first year that I'm completely in charge of it, and it's an absolutely amazing experience.
Nice. All right. Thank you. Alan, what do you have for us?
I have a game. So this is a website. You go there and you can play Bacteria Builder. You get to choose your shape. You can play online. You can also get it on, I guess, the Google Play Store if you have an Android device. You choose what type of shape of bacteria you want to be, what environment you want to live in. You can choose different structures, and the structures have various energetic costs. So you can only spend a certain amount on this, and it'll affect your growth rate. And then it simulates your growth in your chosen environment and determines how well that you did as a bacterium.
This is great. I love
Excellent.
it. I love it. So
give it a try. It was supported by the Society for Applied Microbiology, and this is a free online resource that anybody can have their, any class. This, I think an elementary school class could, like a 5th grade class or up, could probably do this, and certainly any higher level grade.
Capsules stop me drying out and let me stick to surfaces. I guess you're going to learn something here too, right?
Sure.
That's the idea.
Nice.
That's the idea.
You could do this with viruses for sure.
One could. It's probably be a nice thing to see.
I bet
Lots
it's been
of other
done.
things.
This is cool. I really like that. That's neat.
Lots of fun.
Yeah. Rich, what do you have for us?
Yesterday were announced the 2018 Ig Nobel Prize winners.
Okay, then.
So for those of you who are not familiar with this, the Ig Nobel Prizes are a sort of a spoof on the Nobel Prizes. I guess it's a group at Harvard that mostly puts this together. That's where the ceremony usually takes place. So they are, given out by Nobel Prize winners to individuals who've done research that, at least on the surface, seems silly. Okay, but lots of times it turns out that it's not nearly as silly as it seems. So this year there's a medicine prize, an anthropology prize, a biology prize, a chemistry prize, a medical education prize, literature, nutrition, peace, reproductive medicine, and economics, a lot of them. And just as an example, the medicine prize was to a pair of researchers for using roller coaster rides to try and hasten the passage of kidney stones. And the anthropology prize was to a group of researchers for collecting evidence in a zoo that chimpanzees imitate humans about as often and about as well as humans imitate chimpanzees.
Wonderful.
I like the Peace Prize. This paper is shouting and cursing while driving. Frequency, reasons, perceived risk, and punishment. It's quite good.
I like this picture from the 2009.
Yes.
This winner, Dr. Elena Bodnar, demonstrates her invention, a brassiere that can quickly convert into a pair of protective face masks. And there are three guys behind her wearing them. Gosh, it's
a lot of the Ig Nobel Prizes in the past were given out posthumously.
Dickson, you ever get an Ig Nobel Prize?
Hopefully not. I would not know if I did because I was dead asleep at the time.
Very nice. Dickson, what do you have for us?
Well, I have a book that I've just purchased through Amazon called Between Fear and Hope, A History of Vaccines and Human Immunity, which sort of hits all the buttons that we talked about today. It's written by Michael Kinch, who is the chancellor at WashU at the moment, but has had a number of experiences in his life, and he enumerates those in the introduction. He's worked in industry, and he's worked at Yale on vaccine development and that sort of thing. But it really is a tour de force of history. And as a background of this, of course, then he throws in these diseases and how they affected the outcome of history. And then the forward to the present time as to how the vaccine arose to try to counteract this. And he really does a wonderful job. I mean, he's more of an historian than he is an immunologist, I think. And yet he considers himself a molecular biologist by training. And it's quite an interesting, I'm only halfway through, but I'm extremely impressed with the level of knowledge that he's expressing in this book. And it just came out. It was just, it says that there's always been anti-vaxxers. We have never had a time in the history of humanity where if we've had a vaccine or a potential for a vaccine, we've had anti-vaxxers there right away shouting them.
Always there.
Trying to shout it down.
So when the first phones came out, were there people who said we shouldn't use them? They're going to cause brain cancer.
They said, that thing will never fly. Don't get into
Speeds over 20 miles per hour are bad for the heart.
Sure. Yeah. And in some cases, they were absolutely right. The second victim of airplane disaster was Orville Wright. Right? Yes.
I believe that's correct, yes.
Cool. Thank you, Dickson. Mm-hmm. Ned, is there anything you want to share with us?
Well, I don't have anything quite as amusing, but I will. I mean, an area that we didn't get to talk about is related to what we were talking about, but it turns out that VPX does something totally different that turns out to be really interesting. And this is a now evolving story. And really, I think some remarkable papers over the past year. And I'll refer you to one of the, I guess, perhaps the one that kicked it off was a Nature paper. First author is Nian Liu. It's from Stanford University. What they were looking at was these endogenous transposable elements. Mm-hmm. They're like 17% of the human genome. And, maybe they're just junk, but the human genome doesn't want to be overwhelmed by these things. And there are systems that evolved to prevent these things from increasing their copy numbers. And so this paper is uncovering a mechanism that we didn't know about that silences these elements. And it's so the proteins that do it are called the Hush complex. And they cause the histone methylation of these L1 elements.
Cool.
And it shuts them off. And it turns out that HIV, that this system is blocking HIV as well. Since HIV is, looks just like one of these retro elements too. So it is and it appeared in the paper that just came out from the French group, it looks like VPX also functions in order to counteract that silencing mechanism.
So I've got a paper, Liu et al., senior author, Wisaka, selective silencing of euchromatic L1s revealed by genome-wide screens for L1 regulators.
That's the one that identified this Hush complex as turning off these genes. And it turns out that, these little transposable elements, they often sit in introns of genes. So the cell goes and methylates those histones to shut it off. Right. It also affects the expression of that gene.
Yeah.
So these little elements that are sitting in there are affecting gene expression.
Cool.
Yeah.
Neat. All right. Maybe a subject for a future TWiV.
Yeah.
I have a couple of picks that revolve around science publishing. One of them was suggested by Bob, who writes, "You and the profs have been talking about this for years now. I wonder if this will be the tipping point for open access." And he sent a link to a Science magazine article by Martin Ensering. It's entitled, "European Science Funders Ban Grantees from Publishing in Paywalled Journals." Now, a number of different European grantees, which have totaled about 7.6 billion euros on research funding, have said every paper it funds has to be freely available from the moment of publication. No more paywalls. Now, this is not the big European funders, but it's a start. And I think this is really cool that they're going to do this. And I'm just reminded of what the Hughes and the Wellcome did. They just founded their own journal, eLife, with many millions of dollars, but they could have done this, but they didn't want to. Anyway, it's a cool article, and I will also link to the statement from Mark Schlitz, who's the president of Science Europe, and you can see he's got the Coalition for the Realization of Full and Immediate Open Access. So that's pretty cool. That's one pick. And the other is something I ran across while looking at this European thing. It's an article by Stephen Curry. It was published in Nature in February this year. It's called "Let's Move Beyond the Rhetoric. It's Time to Change How We Judge Research." It's basically about not using the impact factor anymore to judge people or the research that they do. It's something we talk about all the time here. And he links to an organization that I didn't know about, which is another part of the pick. It's called DORA. Anyone know what DORA means? The Declaration on Research
Dora the
Assessment.
Explorer.
Exactly what I thought, Alan.
So this is a nonprofit which recognizes the need to improve the ways in which the outputs of scholarly research are evaluated. So it's essentially the same idea, no impact factors. You can sign the declaration. I went there and signed it right away. Many people have signed it. You can read it and so forth. So that's another part of my pick. And then we have two listener picks. One is from Steve. He says, "Since this is an ongoing topic on TWiV, I thought this video might be relevant." And he sends a link to a YouTube video called "Paywall, the Business of Scholarship," where it's all about using the paywall to make money off of our articles. And as a second pick, this book was written by one of the people talked about on TWiV 508, and he sends a link to Ken Alabek's book, Biohazard, the chilling true story of the largest covert biological weapons program in the world, told from inside by the man who ran it. Wow, it's got to be the largest subhead.
Yes.
Yeah.
Steve in St. Louis. And then finally from Islam, a tweet from Peter Suber. "Scholars, spend 68,500,000 hours a year performing peer review. The U.S. federal minimum wage is $7.25 an hour. Scholars deserve more than that, but take it as a starting point. That's a gift to publishers of at least $496,625,000 a year in free labor. And as someone suggested on a previous TWiV, they should give us credit towards page charges when we do free review, right?
Yes.
Because we do it all the time. You know, I remember I'm a PhD
like
student.
miles on airlines.
And Peter Palese says, Vince, I have to review this. Can you help me? And from that day on, I just said, this is what we do. But not thinking, you're doing it for free. So now when they say you're late, I say, hey.
You get what you pay for.
You get what you pay for. That's right.
I'm a latent reviewer.
Yeah. Jeez.
Well, some journals will give you like a year free subscription to their journal if you review papers.
Is that right?
Yeah.
For one paper, you get a year?
I don't know. I think I did for a few journals,
Really?
but not ones that I
Get your
actually
name
I never really read them.
acknowledgments.
I think they should give us a credit towards page charges. So what would a review be worth? Towards, I would say $500 towards page
Words.
charges.
They do it by words.
Words. That's paid by the word. All right. That's TWiV 511. You can find it at Apple Podcasts, microbe.tv/TWiV. Send your questions and comments to twiv@microbe.tv. And if you like what we do, consider donation. You can go to microbe.tv/contribute for ways that you can do that. Dickson Despommier can be found at trichinella.org, the brand new revised website.
Just went up. Yep.
Thank you, Dickson.
You're welcome.
Yeah, so Dickson and I have revised trichinella.org.
Yeah, Vince, you did a great job.
No, I didn't do anything.
No, you did a great job.
Listen to what you told me to do. And it's now a modern website.
It is.
And the cool thing is, Dickson has accumulated all the photos and information he's made over the years on Trichinella spiralis. There's nothing like this site, right? There's nothing like it. And really beautiful pictures.
It's the world's largest intracellular parasite.
Yeah.
Yeah, the largest. You can see it with
The
your
world's
naked eye.
largest.
You can see it's bigger than a Mimi virus.
Really nice. Thank you, Dickson.
You're welcome.
Brianne Barker is at Drew University. On Twitter, she's BioProf Barker. Thank you, Brianne.
Thanks. This was wonderful to be here. I had a great time.
Thought you would like it, being kind of an immunologist, virologist yourself.
Since I think about DNA sensing and HIV, it's right up my alley.
There you go. Great. Rich Condit is an emeritus professor at the University of Florida, Gainesville. He's currently residing in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
Alan Dove is at turbidplaque.com. He's on Twitter as Alan Dove. Thank you, Alan.
Thank you. It's always a pleasure.
And our guest today has been Ned Landau from NYU Langone School of Medicine.
You should mention also that for the first time in the history of the School of Medicine, they are now ranked number 3 by U.S. News and World Report.
NYU Langone?
Yeah, that's right.
What were they before?
They were like 12th or 13th or something.
It was a major jump, but what NYU was in the news for just recently, free medical school.
Correct.
That's right.
Yes.
That was my pick a few weeks ago. That's
right.
Pretty remarkable.
That's right. Yeah.
But why don't they just give it to people who really need it and save the rest?
You know what? I'm going to ask exactly the same thing.
Maybe they could fund some of their researchers with the rest of the money,
Yeah.
We do that here.
You're not the first person to suggest that.
Because not everyone needs it. There are people who have plenty of money, right?
Well, that's
for sure.
That's exactly.
Anyway, thank you, Ned, for coming.
Yeah, thanks a lot. This
We had
has
a
been
great
really, really a lot of fun. Yeah.
Great time.
We'll have you back when you do those mouse experiments.
Okay.
Yeah, Jersey boys are always welcome.
I'm Vincent Racaniello. You can find me at virology.ws. Thanks to ASM for their support and Ronald Jenkins for his music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.